once again, okay, because otherwise most of you are missing out, right? And I'm going to rush through that because these are mostly definitions, what kinds of studies we are going to deal with. Okay, so typically this is biostatistics, you know, right? So we should know what kind of studies we are going to deal in this class, right? So that is what this class is for. Okay, so we are going to define the types of biostatistics studies and what kinds of methods that we are going to apply, numerical methods that we are going to start up in the next class onwards. Okay, so you should know the kinds of studies for that, right? So you know, this is the book that you will have to follow, and for this class is the chapter 2 of Fisher and Belay 2004 book. It's one of the good book, and the book is available in the course website. So some of you might have already gone to the course website. Have you downloaded, any of you have downloaded the book yet? Okay, so you can go and download this book, right? There are 2 books available in the course website, so I suggest everyone to download these books, because these are my own personal copies which I have scanned it for my students. Okay, so I'm going to remove the links in this weekend, so before then you have to download these books if you wish to. So okay, so otherwise also we have so many of these biostatistics books in our library which you can go and take photostats if you like to have it. Okay, so that is what this is, the chapter 2. Well, you know, the observational study, simple. Think about the meaning, word meaning, what observation is, right? We are observing something, we are not manipulating that experiment. So that is what experiment is. Observation against versus experimental study. There is no experiment going on here. We are simply observing the process of nature. Okay, for example, reproduction. You know, you are simply observing in plant reproduction. We are not doing anything. Or if you are a space scientist, with a telescope, you are observing kinds of galaxies and intergalactic matter, or how the planets are moving, all those kind of things. You are not manipulating anything with that kind of thing, right? You are simply observing. So that is what the observational studies are. Okay, so but there are a lot of bias here. For example, all questionnaire surveys are observational. You are collecting the data from the public, for example, right? And you are not manipulating the data, only that you are analyzing the data in context of biostatistics, right? But a lot of questionnaire to the public, for example, I'm asking the public, I'm just going to the bus stand here, Bathinda bus stand, and I'm meeting a lot of public there, and I'm giving a questionnaire. Are you satisfied with for example, health department, the kinds of policies that Punjab State Health Department is making? Everybody is going to say no, we are not satisfied, because we don't have enough doctors here, right? So that is why these are quite biased. The public opinions are quite biased, and sampling errors are quite high in this kind of studies. But if you look at that, okay, there is a special, well, I haven't said that, this versus experimental study. Experimental studies always lead to strong inferences, and that is why most of the scientific studies are experimental study. But that doesn't mean that this sort of observational studies are of not much relevance to the research. There's a number of researchers here. For example, myself, most of my research is concerned with collecting the specimen from the nature. I'm actually working on algae. Okay, so I collect the algae and I sequence these algae to see the diversity of the algae, okay, from the Indian subcontinent. So that kind of study, again, that I'm just observing it, right? I'm sequencing the DNA. I'm not doing any genetic engineering of using that DNA. I'm simply I'm inferring the biodiversity as well as biogeography of these algae. So these are observational studies. Is that clear to you? Right. And there is something called Hawthorne effect, which is quite common for observational studies. If you're observing something and you are manipulating something, for example, in the simplest sense, I'm reducing the light intensity of this glass. Okay, slowly I'm reducing the light intensity with the belief that I'm telling you all. What my belief is that if I reduce the light intensity, your performance is going to increase. So I'm telling you prior hand that this is my experiment. And if I reduce this light intensity and if I observe a good performance, performance is significantly increasing, then that is it's an effect, right? It's an artifact. It's not really that your performance is increasing because of light intensity. If I increase it also, performance can increase. Okay, so your performance is increasing merely because you are under an observation. Okay, so that is a psychological effect, right, or artifact. Are you getting the point? That exactly is as a company in the US that is called Western Electric. They have performed it in many decades ago. That the same thing what I said to you, the light intensity on the working force, or the working output of those employers, that they did a survey of that one. Okay, they did reduce the light intensity and saw that the work output is increasing drastically. Then they increase the light intensity again, they saw the same thing. So if they don't play with that, or if they don't put this kind of pressure, it's not even a pressure, you can say it's just an effect, psychological effect, that the employer's output is increasing merely because they are under an observation, right? So that is the kind of effect what you call Hawthorne effect. Okay, so I have linked up, it's a very interesting story published in Scientific American. It's a blog. Okay, so where you can see some of this cartoon as well as the original raw data from that study, including the photographs taken in the Western Electric. So you go there and read the story. Okay, it's a very interesting story, published only a month ago. That is in August 2013. Okay, so you can have a look at that. Then again, the experiment. What this experiment is? We defined already what observational studies are, right? Experiment or experimental studies, we are deliberately changing some of these parameters to see is it significantly improving or not improving the results. That is what an experiment is, right? Scientific experiment, you must be knowing, right? So many times that we will have to repeat it, right? Triplicate experiment, for example. We are not merely observing the natural phenomenon there, right? We are manipulating some of this experimental setup. For example, pressure and temperature in a closed system. I'm manipulating some of these factors to see the output of it, right? Is it improved or not improved or something like that, right? So experiments normally lead to stronger scientific inferences than do the normal observation study. So that is the difference here, experimental versus observational, right? There are some journals, for example, Journal of Experimental Biology. So these journals deals with experimental studies, not observational studies. Is that clear? That is the difference here, observation versus experimental study. You can say what the kinds of, for example, expeditions of Charles Darwin. So he has been doing a lot of expedition, then analyzing the results of it, right? It's a purely observational study. There is no experiment he did it, right? But still, it's a kind of an experiment, scientific experiment, right? But that it's not actually an experiment where he manipulated with certain factors influencing that experiment. Is that clear? So that is what, right? While observational studies are always open to misinterpretation due to the lack of knowledge in certain disciplines. Okay, so for example, my sort of studies on algae, okay, systematics of algae, for example, or biogeography of algae. If I do something and if I write a big monograph on the diversity of algae in the Indian subcontinent, okay, so if you are not a specialist in algal systematics, algal taxonomy, you don't know, is it really I'm bluffing in my literature, or is it really significant results I'm going to get it, right? That is the problem with the observational studies. For example, Darwin's results, right? So if you are not a keen natural historian, or if you are not a keen person who knows a little bit of evolution, you think that these are nothing, right? The finches, the beak size of finches in different Galapagos islands. Okay, so these have got no significance at all unless you know something about biogeography, something about the process of evolution, right? But at the same time, if a person, for example, a student of this class is performing a study on cancer cell lines effect of 3 different drugs, okay, now you have certain empirical data with it, 3 different drugs, okay, so decrease in cell proliferation rate, and you are performing a special experiment. I'm not a specialist in cancer, but your data I can analyze it to see that is it significant or non-significant. I can compare the means, and I can perform something like t-test, student t-test, to see 2 different means I can compare. If it's more than 2, the whole together, I can perform an ANOVA, simple ANOVA, to see is it significant or non-significant. If at all there is some differences in the mean, okay, the average, if there is some differences in the average, I can perform it. Or first of all, I can see is average same or average are different. Okay, so that is what it is. So observational studies are prone to this kind of error, while experimental studies are a lot more robust in that numerical sense. Okay, so it's clear? Right, what is this comparative experiment? Again, you should know the meaning of the word comparative. Here we have a department of comparative literature. What they are doing? Yeah, they are comparing, right? Comparing different kinds of literatures. It's the same way, experiment. You are not doing one experiment. You are doing a battery of experiment, and then you are comparing the results of these experiments, right? So that is what the comparative, right? It compares two or more techniques or treatments or levels of variable, level of a variable, in a random variable. For example, drug level, right? So, or in the previous example, light intensity is a variable. You're looking at the effect of that random variable, and to see that is your experiment is differing or not, the endpoint or the result of the experiment. Okay. An example is treatment or versus without treatment. So without treatment could be a negative control, or it could also have a positive control. If you are looking, for example, a mutagen, okay, mutagenic effect or genotoxic effect, you can have a non-DNA mutagen, for example, ethidium bromide, as your positive control. Okay, your negative control could be a milk water, pure water, no mutagen at all, everybody knows it. And your experiment could be water samples taken from the hostel. Okay, so these three things you can do that, and you can compare it. So that is a kind of study of what you call it as comparative experiment, right? You are performing an experiment, you are comparing the results of these experiments, right? Then you know what the experimental unit is. It's nothing but the smallest unit on which the experiment is performed. For example, if my experiment is to improve the standard of the students, my students, experimental unit could be the class, the whole class. It's not a student, a one particular student. If one of you student, the performance is increasing, that doesn't mean that my class is actually I'm improving myself, right? I need the whole class average to be increased. Is that clear? So that is an experimental unit, right? For mostly for epidemiological studies, human beings, human, right? And it's not just one patient, it could be a cohort, a group of patients. Is that clear? That is what experimental unit is, nothing, right? It's a very simple definition for that. Then there is another called crossover experiment. What is this crossover? It's the same unit. For example, a mouse named X. Okay, the same mouse is getting treatment with the drug A. After that treatment and after that experimental, we got that experimental data. Then subsequently, the same rat undergoes treatment with drug B, and after that treatment with drug C, in the sense that there is no overlapping time, drug A then drug B, so in between 2 days drug A plus drug B, no, there is no such overlap. So that is a set in the logic, there is no union between these two sets. Okay, it's a non-overlapping, right? So that is what a crossover experiments, right? So investigate under more than one condition of the experiment during non-overlapping time periods. Okay, so sequential treatment of lab animals with various drugs and blood metabolites are measured with each drug. So this is an example of this sort of crossover experiment, right? So what the cohort is, mostly with the people, right? Human beings, we call this term cohort. So it's a group of people whose set membership is clearly defined. Okay, so if I say my cohort is smoker, so I have to define who a smoker is. So I now define the smoker is an individual who smoked more than one cigarette in the last year. So that means if a person who smoked one cigarette last year, he is not my cohort. Okay, so I define who my cohort are before performing my experiment, because that definition is a very important, right? If I erred in this definition, then my whole experiment is erred. Is that clear? So all students enrolled in MPhil PhD program of this university is my cohort. So it's a very clearly defined, right? So definition should not be ambiguous. So if it's ambiguous, then it's not a cohort. You cannot say that way. Okay, so most of these epidemiological biomedical studies, the cohorts are followed temporally. Temporally means what? Temporal means time, right? For a course of time period, for example, 1 week or 3 months or 5 years, right? We are following temporally. And what is the spatially means? Spatially means from one space to another space. For example, Punjab, Kerala, or Delhi. We are comparing these 3 states. So that is what spatial follow. Okay, spatio-temporal means both factors, space as well as time that we are taking in. Is that clear? Okay, so what is this endpoint? Endpoint is nothing but an end, a point where the experiment is deemed to have completed, right? I'm sure everybody might have performed in your high school or at least in plus 2 some practical on chemistry, right? The volumetric titration, right? So acid base titration where phenolphthalein or some kind of indole, for example, those indicators we are adding and we are titrating with burette, right? And there the end point is change of color of the indicator that causes change of color, right? With the pH, of course, right? In the acid base titration. So that kind of titration we call endpoint titration, where endpoint is change of color, right? So that kind of endpoint, presence of a disease. So that could be an endpoint, right? Or 5-year survival period after radical mastectomy. So mastectomy is nothing but removal of breasts. Okay, one breast or both the breast because of the the breast cancer, right? Mostly. So are they surviving or not? We don't know. 5-year. If it's 5-year survival, that is my endpoint. 5-year survival. If a patient, a cohort, is dead within 5 years, then that is not my endpoint. I'm not going to take that cohort in my study. So only those patients who are surviving after mastectomy for 5 years after the radical mastectomy is my endpoint. Or death, mostly many of these epidemiological studies, death is the endpoint. Patients are followed till the patients are dead. Okay, so there are 2 kinds of study here, prospective and retrospective. Again, it's just a word meaning, right? You can just know what meanings of the word prospective. It's nothing but prospectives, right? So your university prospective, what we are doing, what we are going to do, what we are going to achieve, it's a future, right? So cohort is followed for the occurrence or non-occurrences of the endpoints. Okay, so particular patients, for example, some patients in the general public, we are selecting these patients, HIV patients, and we are following the development of the AIDS in this HIV. Most, not all HIV positives develop to AIDS, right? Some of them develop to the AIDS. So we are following temporarily. That means 5 years, 10 years, till they develop the AIDS. So what are the kinds of features, what they are showing, or those kind of things are what you call prospective. So we are taking it and we are following it temporarily, right? It's a futuristic study. Is that clear? While retrospective study is not futuristic, but it's a retrospective. Retrospective analysis means I'm thinking about the past, like a post-mortem, right? So I think what would have been caused this disease. Today a particular person is having HIV. So I'm investigating what would have been caused this particular person HIV. That kind of retrospective study. Okay. So most of these genetic studies are retrospective. So we are taking the gene data, the sequence data, or the cytogenetic data, okay, and then we are looking at that the factors that would have been contributed to the development of that particular condition. Or it could be prospective as well. Angelina Jolie's case, right? The BRCA1. So a particular set of SNPs in that disease, that gene, are associated with breast cancer risk. Okay, higher breast cancer risk. That's all about the probabilities we are going to cover in this class. What is the probability of developing a particular condition? Okay, so that is a retrospective. Is it retrospective or prospective? It's a prospective, right? Because the particular person didn't develop that particular disease yet. We are looking at the probabilities of developing. Okay, so that is what prospective versus retrospective. Okay, so use of cancer registries or twin registries, so to compare causal factors for that particular cancer. For example, Bathinda area or Mansar area of Punjab is prone for breast cancer. Cancers, and among them breast cancer probability is very high. And now I am set to investigate this matter. Okay, the place where I can start up is I will look for the cancer registries of the hospital and look at that patients, and I'm now looking at the factors contributing. Is there any work related factors? Okay, so is it persons who are working in agricultural fields more prone to have cancer, okay, or breast cancer in particular? So that kind of thing I have to look at the cancer registry. Unfortunately in India we don't have very good maintained registries. That's the main problem. If we had it, it would have been much easier for the researchers like you to perform this kind of study, retrospective study or twin study. So mostly Scandinavian countries, that is the Northern European country, where they have excellent twin registers. So we can say if it's a homozygotic twin, and a particular twin is getting this disease while the another twin is not getting, what would have been occupational association for development of this particular disease? That we can see that easily looking at those registries and looking at the cohort. Those cohorts are twins, could be heterozygotic or homozygotic, right? Okay, so case control study. What is that case control study? So it study, this is the kind of study that selects all cases, then controls as well. So a case plus control. Case is nothing but patients of a particular disease, and normally controls are healthy individuals, and we are looking at these two things to see the inference of the particular thing we are in question of, right? So case control study have case and then control, and control are mostly healthy individuals. Okay, so group normally called control, often negative, who are healthy patients and serve as comparison to the cases is also selected. The cases and controls are compared with respect to various characteristics. Is that clear? It's very simple. Case control study. Okay. For example, a study on dietary factors contributing on the development of coronary heart disease. Okay. So in that case, I'm selecting heart patients as my case, and healthy individuals who don't have pulmonary heart disease, coronary heart disease as my control. And then I'm looking what kinds of dietary habits that these two groups have. Okay, is it a high fat contributing to the disease, or is there any association with consumption of alcohol, for example, smoking, for example? It's not dieting, right? It's all behavioral factors, in fact, if I include smoking as well. So that is what the case control study. What is this longitudinal study and then cross-sectional study? Again, longitudinal, if I say in the graph, you look at the longitudinally means look at across it, right? So that is what the what is that? Latitudes and longitude. Latitudes and longitude, right? So that is what it is. Longitudinal study collects information of study units of a specified time period. So information on study units, mostly cohorts, patients. We are selecting some patients here, and then we are following temporally. 2 weeks, 3 months, 5 years we are following, and that is what we call it as longitudinal study. Is that clear? So followed temporally means that this patient I select here, and here the x-axis is time and y-axis is nothing but names of the patient, for example. Okay? Patient A, patient B, patient C, like that. So here this patient I select here, and I start here from January to August, and this is February to September, like that, right? So I'm following one patient over a course of time. That is what we call as a longitudinal study. And what is this cross-section study? Cross-section study means I'm not just looking at one particular patient, I'm looking everyone on a particular time. Normally it is a day, right? One particular day. For example, today, the 23rd of September 2013, I'm doing a HIV test, for example, ELISA test in the bus stand, and to see how many positives I'm getting, how many negatives that we are getting. And that kind of experiment is what we call cross-sectional study. Is that clear? So we are sectioning here on a particular time. Okay, it could be one particular time of a day, or it could be a day, or it could even be 1 week time. Okay, so anything, right? A time you're defining, and then data collects on a fixed time. That is what a cross-section studies. Then what is this placebo? Placebo effect or placebo treatment? Something like Hawthorne effect. Hawthorne effect is it's a psychological effect, right? Effect only because that people know that they are being observed, right? So placebo is something like you're going to a doctor. You think you have a particular ailment which is very serious. For example, you have a heart attack, and you're going to a doctor, and doctor knows you don't have a heart attack, but because your psychological effect is such a strong that you really have heart attack, and if doctors say you don't have heart attack, you will disbelieve him or her, and then doctor prescribes some medicine, for example a vitamin, and then this vitamin capsule, the doctor say this is a very important cure for your deadly heart disease, you have a heart attack, okay. And this particular tablet will cure all for all of your ailments. So that's nothing, it's a plain lie, right? It's not a truth at all. It doesn't have this. Placebo have got no physiological effect on the patient, it's only psychological effect, right? So because the patient believes that this is going to improve his or her health status, so that kind of effect is what we call placebo effect, and that kind of treatment is what we call placebo treatment. Is it clear? Then what is a single blind versus double blind? So that term also you should be aware of, right? If subjects being treated are unaware of the which treatment, including any control, that they are receiving, that is what you call single blind. For example, a doctor, okay, doctor prescribes, mostly this homeopaths, right? It's not science, right? Homeopaths, what they normally do is that they give you some medicines. Every medicine looks exactly same, right? White ball or whatever, right? So this sort of thing, and if, right, most of the homeopaths, I'm not criticizing them, but they mark the bottles with the particular treatment, right? Whatever the treatment they are administering. But in case, let's say a particular homeopath, he is not marking anything on the bottles of the treatment what they are administering, and then the patients do have no idea what this medicine is, right? Because the doctor supplied a blank bottle with no label, but with medicine. So that kind of thing could be something called single blind, where the doctor knows it, okay. But double blind means even doctor do not know, right? Doctor gives the this is the particular problems that the patient is having, and he refer that to a druggist, for example a chemist, because doctors are not too good with the drugs, right? Especially in the West, doctors consult with the chemist while deciding which drug to treat, because of the drug interactions that doctors are not really aware of, okay. So in that cases, especially in the case of some complex disease where the patient is getting some medicine already, okay, so the doctor refers that particular patient without revealing his or her true identity to a chemist, and then chemist prescribes a color coded or some coded medicine to the patient, in a sense that patients do not know the identity of the medicine, neither the druggist knows the identity of the patient. So that kind of thing is what you call double blind, that both of them do not know. Is that clear? It's very simple. We in this class we have simply defined certain the kinds of experiments, right? Various kinds of experiments that we are going to deal in this class, okay, differentially for most of these studies. For example, in the quiz I can ask you, in a double blind study you should know what this double blind means, right? Just a definition, and I suggest everyone to look at that particular chapter of Van der Leeuw book, chapter 2, and accustom yourself with other things as well, right? I only defined some very important terms of different kinds of studies, okay. Is that clear? Now let's go for a quick review, okay. Some questions. Where are your attendance sheets? Have you completed it all? Can I have that? Okay, thank you. Right, the first question here is to improve learning abilities of students in my class, right? I implement a policy that all students use green ink in their notebooks. I see a significant progress in class grades. What would be the possible explanation for this improvement? Okay, so can Ankush Pandey answer this question? Yeah, can you answer this question? It's a question like, what would be the possible explanation for the improvement of the students' performance? If I ask my students from tomorrow onwards, all of you have to use green ink, green pen. Okay, then after 1 week I conduct a flash quiz, and I see that today's quiz the performance is really significant. Okay, to see that is it really significant, I have to perform something called student t-test, dependent t-test. Okay, because each student today and each student after 1 week I have to perform. So what do you call this kind of study, or the effect due to this policy? It's an observation study, that's right. And what do you call this kind of effect? There is a particular fancy name for that effect. What is that effect? H starts with H. Do you remember? Hawthorne effect, right? So that is what the effect, this kind of effect. In the, I told you, in the Western Electric, exactly what they did is they played with the intensity of light to see the work. Okay, so the work output, is it increasing or decreasing? Only because they are under observation, they know that someone is looking down, like big brother is watching you, whatever you are doing, right? So their performance is increasing. It's a psychological effect, right? Sit down. So that is what it is. Is that clear? Okay, now in a cancer therapeutic research, a particular breast cancer patient was identified and subjected to 3 weeks treatment targeting X pathway intermediate. These are targeted therapy, right? These days mostly cancer therapeutics are targeted. They are targeting one particular pathway intermediate, right? And after that, subsequently, okay, the patient was subjected to 4 weeks treatment targeting Y, another pathway intermediate. How would you call these kinds of studies? And I asked this question to Amrit Pal Kaur. Yeah. You don't know this. Think about it. What would that be? See, one catchword here is a subsequently, right? Subsequently means that there is no overlapping time period. So the same patient, crossover. Yes, that's right. So this is what you call the crossover, right? Crossover experiment. Sit down. Is that clear? Crossover experiment. It's very simple, right? Now my next question here. Upon talking for a couple of minutes, doctor identified Mr. Gopal Iqbal John, okay, so a doctor's fictional name, as a case of typical hypochondria and prescribed a cobalamin. Okay, so he prescribed something called cobalamin. Patient asked, what is this for? Doctor replied, this is a new generation cure-all drug, which would surprisingly cure Mr. Gopal's all problems in next 2 weeks. What would you call this kind of treatment? Just wait, right? Don't say that out. So I'm asking this question to Al-Karani. Okay, placebo effect, right? Good, excellent. Sit down, please. So here it is. Can you answer what this cobalamin is? B12. Yeah. Is that clear? It's just a vitamin, but the effect here. Now, the effect is called the placebo effect, right? Now, next question here. Don't say out loud, okay? So I just want to see how you are performing. Scholarly scientific communications are generally subject to the process of peer review. Scholarly scientific communication means your articles that you are putting in the papers, right? So, in which upon submission, editors forward the manuscript to 3 or more reviewers with the identity of authors suppressed. The reviewer will not know who these authors are, okay? After peer review, editor contacts the authors with the comments from reviewers, again with their identity suppressed. What would you call this sort of review process? Is that clear? So only one person knows it, the identity, that is editor. Editor knows everything. But if you submit a paper to the editor forward that paper to 3 different reviewers with your identity concealed. Okay? And after the reviewers comments back, the editor forward the comments of reviewer to you. Again, identity of these 3 reviewers concealed. So only one person, the editor knows everything. Okay? So now I want Shefali Singh answer this question. Who is this Shefali Singh? Yeah. Huh? Single blind. Single blind means only one person knows it. Here in this case, it's both of them do not know, right? It's double blind, right? It's not a single blind. Is that clear? Single blind versus double blind. In case, sit down, sit down. In case the editor reveals the identity of the original manuscript author to the reviewers, then it would have been single blind. Because you don't know who those three reviewers were, but reviewer knows who you are. Okay? But here both of you do not know. Reviewers as well as the author do not know each other's identity because everything is concealed. That is why these are called double blind. Is that clear? Now, Public Health Department of Punjab conducts a sporadic flash HIV test on 23rd September today. The same thing, I put this example here in this today's class, right? Across Punjab, what would you call this kind of experiment? So experiment on a particular day, it's kind of a flash HIV test. Anuradha Kumari, please. Yes? Case control. Where is the case and where is control? There is no case and control. It's everything is all about general public, right? I'm not defining who the case is, I'm not defining who the control is, right? And case control study have to follow temporally. Case I'm defining, control I'm defining, and I have to follow temporally. For example, a course of 2 weeks. Okay? And what is this? It's a cross-sectional study, right? Performing because one particular day I'm looking at different individuals, and then sit down. Is that clear? Okay, no ambiguities, right? Now my next question here is 56 patients who were tested positive for HIV-1, type 1, were selected and were subjected to extensive counseling to study the behavioral factors that might have caused the disease. What would you call this kind of studies, right? Rashpal, please. Yeah, Rashpal. Prospective. Prospective is like, what are the prospects of this experiment? Means future, right? Is it future or is it past? Yeah, it's retrospective. Is that clear? Because I identified a particular person. Okay, sit down. A particular person is identified. I'm asking him in the counseling, what are your behavior? Okay, so that would have contributed in HIV. So I'm cross-questioning. It's something like a police station, right? So what police guys are doing after a crime, right? So they identified some culprits, and I'm asking, what did you do at that particular time? So all about past. Also in the forensics, right? A particular person is it's something like a murder victim is brought here in my hospital, and then I'm performing postmortem to see the cause of the death. Is it murder or is it suicide? If it's murder, what would have been caused that kind of death, right? Of course, right? So that kind of studies are all retrospective, right? But I'm not thinking about future there. I'm looking at the past factors that might have caused that particular disease, right? Another question here, further research on MDR tuberculosis concentrated on 77 cohorts who were temporarily followed 6 weeks for the disease prognosis. What would you call this experiment? And I'm asking this to Nisha Bansal. So I have defined these are my cohorts, right? And I'm following temporarily, that is 6 weeks I'm following it, okay, for the disease prognosis. Okay? And then what do you call this one? I define that, is it a kind of a prospective study? But I'm following that, right? I'm following for 6 weeks for the disease prognosis. I haven't defined what the endpoints are. Normally for a prospective study, you have to define the endpoints. Cross over, right? So then what would be this one then? Longitudinal study, right? Because you are following that particular cohorts for a certain period to see the thing, right? What is this MDR tuberculosis? Anyone? Let's sit down. resistant, right? MDR. So that is what we are looking at here. Now my next question here to study epidemiology of dengue fever. 50 dengue fever patients and 50 health subjects were selected and studied for 3 weeks. Okay, what would you call this study? I'm asking to S.K. Gupta. Again, there are two kinds, right? I'm dividing my group to two. That is, one is dengue fever patients, and then another is 50 healthy subjects. So this is a case control. Is that clear? Right, that is what it is. It's a case control, which is an example of a comparative study. So that is, again, it's a comparative. I'm comparing cases versus I'm comparing the control, which is a positive control in this study. Okay, so that is comparative as well as that is case study. Next question here. To study epidemiology of chikungunya disease, patients were followed temporarily till they recover completely. Okay, and then what would you call this study? I'm asking to Iqbal Gill. Till they recover completely. So patients were followed temporarily till they recover completely. So it's an observation, yeah, of course. And the end point here is what? Till they recover. Yeah, till they recover completely is my defined end point. Okay, so I know the end point. Now I'm following some cases temporarily till they recover completely. What would you call this study? It's a prospective, right? It's not retrospective, but it's a prospective. And we don't know when they are going to recover because it's in the future. It might take some 1 year probably, or even 2 days he can recover, right? So that is what the prospective study and retrospective, right? Now another question here. In the Republic of El Paradiso, doctor prescriptions are all coded by set of numbers. Okay? Identity of which only registered chemist knows. So chemists supply the medicines in blank covers, such that patients wouldn't know the true identity of the medicines that they are having. What would you call this particular study? So I'm asking this to Wahidul Rahman. Yeah. Single blind, that's absolutely correct. Okay, sit down. Because the doctor knows it, right? And even chemists know, but the patients do not know the identity of it, right? That's why. Now this is what research methodology which you are attending, right? Look at that class called experimental design, as well as these sections of the the chapter number 2 of the Fisher and Van Belle steps of the study, ethics of study. Ethics are very important. I'm not covering here because the scope is too different, right? I'm actually dealing with biostatistics, right? Collection, how do you collect these things? Editing the data handling, sample size, inferences, all these sections. Look at that chapter, download the book and study it yourself. Clear? All right, thank you.